All right. Chen from Bates College in Maine, is rising junior working in Keene's laboratory.
All right, hi everyone, thanks for coming. I'm Karen, and today I'll be presenting on the use of digital image analysis to assess Alzheimer's pathology in the visual cortex. So just to begin, a brief overview of Alzheimer's disease. It typically affects about 1 out of 3 seniors over the age of 85. It's characterized by symptoms like memory loss, problems with judgment making, and changes in personality. And as we're all living longer and America is getting older, Alzheimer's is becoming more and more of a public health issue, especially since, well, there's no cure for it. Additionally, even if a person starts exhibiting the cognitive decline we associate with the disease over the course of their life, it's only until a post-mortem autopsy by a neuropathologist that they'll be officially diagnosed with Alzheimer's. So in addition to the general brain atrophy you see over here, when the pathologist looks at a slide of brain tissue under the microscope, they also look for the presence of 2 important proteins, amyloid beta and tau. In Alzheimer's, these 2 proteins start aggregating abnormally and forming what we call amyloid beta plaques and tau tangles. Now, tau and amyloid beta start the accumulations in different parts of the brain, amyloid beta in the outside and the cortex moving inward, while tau starts in the transentorhinal cortex, way inside the temporal lobe, and then moving outwards towards the frontal, parietal, and then finally the occipital lobe. A pathologist will then use the location of these protein aggregations to stage the severity of the disease. Tau has 2 staging systems, Sarah and Brock, but I'll just be talking about Brock for the sake of this presentation. How Brock staging works is that it's location dependent. So if I had a brain that only had tau tangles in the transentorhinal cortex, I would just stage it as Brock stage 1. If the tangles have moved all the way up to the hippocampus, I would then move it up to Brock stage 2, so on and so forth. Now, the problem with Brock staging is that it's only semi-quantitative, meaning that it lacks a lot of nuance. For one, it completely ignores the presence of an earlier form of accumulation that we call diffuse tau, which you can see over here. It's stained a lot more lightly and it's more spotty compared to the more mature tangle. So if I had a brain that was Brock stage 2, meaning that it had the tangles up to the hippocampus, if that brain also had diffuse tau all the way in the occipital lobe, unfortunately, we'd still only classify it as Brock stage 2, despite the tau having migrated to a completely different part of the brain. Secondly, Brock staging is only location dependent, which means it doesn't tell us much about quantitative information. So if I had 2 brains that were both Brock stage 6, meaning that the tangles have gone all the way up to the neocortex, one brain could still have drastically higher tau burden compared to the other brain. And yet, because Brock staging is only location dependent, those 2 brains will still only be classified as stage 6, despite their quantitative differences. So this is where Halo comes in. Halo is a pretty recently developed digital image analysis platform, and it allows us to provide more quantitative analysis of a piece of tissue than what's currently possible with just, well, my eyes, a microscope, and Brock staging. It could do things like count the number of inhibitory interneurons on the slide, the amount of a certain kind of interesting protein, or the co-localization of 2 proteins. This summer, I helped with my lab's Department of Defense study where we measured pathological tau levels in different brain regions across Alzheimer's stages. I specifically helped run Halo analysis on slides containing tissue from the cognitive cortex over here. So the cognitive cortex houses our brain's visual cortex, which receives information from our eyes. The cognitive cortex is located in the occipital lobe, and the occipital lobe is a pretty interesting region in Alzheimer's research. Like I said earlier, it's a loss-affected region by both the tau tangles and amyloid beta plaques. However, there's also been recent interesting studies that show there might be a relationship between changes in our eyes and the progression of Alzheimer's disease. And as such, there also might be a correlation between these changes in the cognitive cortex and changes related to Alzheimer's. So to investigate, we sampled the cognitive cortex like so, embedding paraffin created slides. We then use immunohistochemistry to stain those slides for pathological tau or phosphorylated tau, and use hemotoxin as a counter stain for cell nuclei. We then scanned and uploaded those slides onto Halo, where we then ran an error quantification algorithm on the occipital gray matter. They'll tell us the percentage of the tissue that contained the pathological tau protein. So this is just an example of what the slides look like on Halo. These blue dots over here are the cell nuclei, which have been stained by hemotoxin, and the brown splotches are the pathological tau. And here are our results. I just have a bunch of dot plots and showing data from the temporal lobe, frontal lobe, and finally occipital lobe. X-axis we have Brock stage, and Y we have percent of tau-positive tissue. As you might expect, when you increase the Brock stage, there's also a corresponding increase in the level of tau we see per slide. However, what's interesting is that in these really early Brock stages, 1, 2, 3, where we wouldn't expect any tangles in the occipital lobe, there's still a fair amount of pathological tau. Even more interestingly, the level of pathological tau is quite similar to the tau burden we see here in the frontal lobe in these early stages. The frontal lobe is affected way earlier in the course of Alzheimer's disease compared to the occipital, so we actually might have expected it to just have a higher tau burden compared to the occipital, just as how the temporal lobe, which is the first affected lobe, has a higher tau burden compared to all of these. I ran a one-way ANOVA with the postdoc Tukey test on the average percent of tau-positive tissue for the occipital and frontal lobes, and found that interestingly, there wasn't any significant difference between these two regions. Moving on, I took the data from the occipital lobe and created a box and whisker plot. As you can see over here in box stage 5 and box stage 6, there's just a huge amount of standard deviation compared to the mean average tau-positive tissue, which just goes to highlight the problem of box staging and its ability to provide us more quantitative information about a piece of tissue. Future directions. One of the issues is that we didn't have as many slides with brains that are box stage 0 or 1 compared to the later stages. So hopefully in the future, as we continue to receive brain donations, it might be possible to complete halo analysis on those more representative brains. We hope to correlate tau burden with a more general staging system called Alzheimer's disease neuropathologic change. And we hope to compare the ratios of diffuse tau burden versus tau tangle burden in the occipital and frontal lobes. And that's the end of my presentation. I'd just like to extend a huge thank you to everyone involved in the NSSP program over the course of the summer, Dr. Elbogen, Dr. McDonald, and everyone in the program selection committee for selecting me to be part of it. Thanks. I also want to thank everyone in the Brain Lab, Dr. Keene, my PI, for letting me join, Dr. Bradley Wacker, and the histology team downstairs for teaching me about cell culture and brain anatomy, Victoria Radcliffe, Dr. Nadia Pusatna, and the entire QuantPath team for allowing me to join their exciting projects. I'd also want to thank Bahari and Isabella and my fellow NSSP summer interns for their lovely advice and support over the summer. Thank you so much for coming.